Right, thank you very much for joining us for our talk tonight, which is part of the Breaking Ground exhibition event series. So the Breaking Ground exhibition gives us an insight into the birth of UK paleontology in the 19th century through the lens of the work of William and Mary Buckland. And it uses some of our most exciting and exquisite archives and fossils that we have in the collection. So if you haven't seen it, please do go and have a look before April 2026. But tonight, as part of the series, linked perfectly to it, we're lucky to be joined by Professor Emily Rayfield. So Emily did her undergrad degree here in Oxford before moving on to a PhD in Cambridge, and then postdoctoral positions and fellowships in Oxford, Cambridge, and the Natural History Museum in London, before eventually settling in Bristol, where she is now the Professor of Palaeobiology at the School of Earth Sciences. So Emily uses computational engineering techniques to explore the relationship between the form and function of animal skeletons. Her name is synonymous amongst paleontologists and wider with dinosaur cranial biomechanics, some very exciting work, but her work also expands to cover avian dinosaurs, birds, mammals, marine reptiles, early tetrapods, sponges, algae, bivalves, forams, and excitingly, scaphopods. So this work has earned her an illustrious scientific awards, most recently the Gabor Medal from the Royal Society. And then Emily is also featured as one of our talking heads in our exhibition. And returning slightly to the themes of the exhibition, which includes some fantastic examples of 19th century women's contributions to science, Emily has been and continues to be a fantastic role model for young women looking for a career in science. I hope you don't mind me saying. I know from personal experience as a former student that her work has inspired us all to create, support, and communicate exciting research to the best of our ability. So, thank you very much. Please take it away, Emily.
Thank you ever so much, Kelly, for that wonderful introduction. It's really great to be here. This was one of my favourite places to come when I was an undergraduate and just wander around the museum. And it's a real honour, actually, to be featured in the Breaking Ground exhibition at the moment as well, too. So as Kelly said, the work that I do involves looking at extinct animals, but trying to understand how they actually existed, how they interacted with their environment. And my work particularly focuses on looking at feeding as well too. So what I'm going to talk to you about tonight, I thought would be an interesting topic, would be how do we really know what we know about the fossil record, and particularly kind of with a view to, I mean, all of you have watched paleontology documentaries, and the talking heads, including myself, have said, this happens, and this happens, and this happens. How on earth do we know this, and how do we, imbue some kind of certainty in those comments that we make? So the kind of overarching theme of my talk really is kind of, how do we know that? And so I thought I'd begin with a recent documentary, that a little snippet from this that I was very fortunate to be involved with, where you'll see me appear right at the end and make a grand statement about the behaviour of this extinct animal. So I'll start this now. It runs for about a minute and a half. So there we go, cue suspenseful music, and then me at the end saying that that animal had a bite force that was twice the force of a great white shark. So how do we get to that kind of point, at that level of me being certain enough to go on national TV and tell Sir David Attenborough that exact number? So that's kind of the gist of what we're going to go over today in the talk. So understanding fossils obviously starts with the fossils themselves. This is the actual fossil that documentary was based on. It's a beautifully preserved skull of a Pliosaur, an extinct marine reptile that was around in the Mesozoic, as it said on the documentary, in the seas of the Mesozoic while the land was dominated by dinosaurs. It's beautifully preserved, and using this we can tell a good deal about the lifestyle and the behavior of this animal. It's enormous. It has huge teeth. It's going to be one of the top predators in its environment. The teeth are large and robust. They were capable, presumably, of dealing with quite a lot of stress during feeding. And it has these big openings here at the back of the skull, which we know from looking at living animals would have been packed full of muscles that close the jaw. So using the bones themselves, we can tell a certain amount about animals in the fossil record. Sometimes we get really lucky and we find direct evidence. So the next two slides are from really beautifully preserved fossils that are from the early Cretaceous of China, This animal here, Sinocaleopteryx, is a smallish dinosaur. It was about 2.5 meters in size. It belongs to the theropod dinosaurs, which you're probably more familiar with as being the group that includes large animals like T-Rex. This fossil was preserved with some material in its gut region, and when this was looked at in detail, it was found it was not just one but two different types of animals were preserved in here. In red here we have some long bones from the leg, which are diagnostic enough to tell us that they belong to the leg of another dinosaur. But also this blue coloured mass here actually represents a mass of feathers, which, based on this and other bits of evidence, suggests that this animal also had just eaten a very early bird as well too, an animal called Confucius ornis. So this is another animal here. It looks like a bird. It's actually from a group of dinosaurs that are quite closely related to birds. It's called Microraptor. It's famous because it has feathers on its forelimbs and on its hind limbs as well. This animal was probably about the size of some of the large kind of red kites that you see flying around in the Oxford-Oxfordshire area. And in this, there are hundreds of specimens of Microraptor, but particularly inside this specimen here, again, there were some unusual bones preserved in the gut cavity. And when we look at those in more detail, they resemble a foot. And they're a very distinctive foot because they have an ankle bone structure that's only found in mammals. And so in the belly of this animal was the foot of a tiny mammal, as you can see in this little reconstruction here. So sometimes we don't get the remains of animals in the gut cavity, but we do sometimes get bite marks or teeth that have still been embedded into the prey. So here's a tooth of a predatory dinosaur that's found its way embedded into the vertebrae of a large herbivorous dinosaur here. So that gives us an idea of these predator-prey interactions. Sometimes we find these coprolites, fossilized feces. So this was a fantastic discovery from a number of years ago from the late Cretaceous of North America. When it was published, it was called a king-size theropod coprolite. And the name refers to the fact that it's enormous, it's about this big, and it also refers to the fact that the king refers to the T-Rex, Tyrannosaurus rex, which is probably the animal that generated this fossilized feces here. Now this is interesting even just in itself, but actually the authors of this paper chopped it up, looked inside, and found it was full of pieces of shattered bone. So that gave us a clue, obviously animals eating bones, but because they were shattered and they were quite angular still, they hadn't spent much time in the gut cavity being digested by stomach acids, and it appears that they passed through quite quickly. So that tells us something interesting, not only that the animal's eating other dinosaurs, which is kind of what we would predict, but also a bit about the physiology of the animal, like it had quite high throughput through its gut. And if we return to classic comparative anatomy, the shapes of the teeth actually tell us something very broad about what animals were eating. These curved, sharp teeth here that we find in animals like snakes, and this monitor lizard here, are obviously classic indicators of animals that eat meat, that are using their teeth to slice through prey. This is another lizard here. It's an iguana, and it has, particularly on its upper jaws here, these shaped teeth, which we call leaf-shaped teeth. This animal eats plants, and we find leaf-shaped teeth in the jaws of many other animals which we think are herbivorous dinosaurs. And you can see some here, for example, on this slide. So that's great. So the bones and the teeth can tell us something about how these animals, what they might have been feeding on. And these are like the kind of the hard parts of the animal's body. But what we want to really understand to understand how these animals feed are the mechanisms that move those hard parts. So that will be things like the muscles and the tendons and the ligaments as well too. And obviously, as you all know, animals that fossilize, the last thing that tends to be preserved are soft tissues like muscles, like tendons and ligaments. So how can we go about, in a kind of guesstimate-y kind of way, understanding a bit more about these soft tissues in fossils? Now, one of the things we can do is look at living animals and the closest relatives of some of the fossils that we're interested in. So for dinosaurs, as Kelly mentioned in her introduction, we know that birds evolved from within dinosaurs. We can look at birds, and indeed I've spent many times looking at the soft tissues in birds. But the thing with birds is that they evolve a big brain. And what that does is it shifts the position of their jaw closing muscles and makes them a lot smaller as well too. And they have some rather neat ligaments in their jaws that help move the jaws that we don't see in evidence for in extinct animals like non-avian dinosaurs. On the other side of what we would call the kind of extant phylogenetic bracket, which is just another way of saying like the two living groups that bracket the extinct animals, on the other side of that we've got the crocodiles. And crocodiles have a skull structure, and for what we know from studying them, a muscle arrangement that looks quite similar to dinosaurs. So we can dissect animals, this is a number of years ago, we were dissecting this alligator skull here. And what that enables us to do is get an idea about where the muscles are positioned in the back of the skull, the muscles that close the jaws, how big they are and how big they are relative to each other, their orientation, so what we call their line of action, which influences the leverage that those muscles have. And also we can measure their mass, and we can look at the individual fibers or the bundles of fibers that constitute the muscle. And they're important in telling us how much force that muscle could generate. So we can get all of that information from closest relatives, living relatives of our fossils. We can then use those dissections to map out where different muscle groups attach to the underside of the skull and to the jaw. We can then take this information and look for those same kind of clues of attachment points in fossils. And indeed, this is what we do. So where these muscles attach in the living animals, sometimes they leave a scar, sometimes there's a ridge that delineates where two muscles come together and attach. If there's a tendon, there might be a really obvious rough surface where that tendon is attached. So we look for these clues in fossil bones, particularly at the backs of the skulls. This is a fossil crocodile, so unsurprisingly, when we look at this, we think its muscles look quite similar to living crocodiles. But the same can be said of dinosaurs as well, too. They have quite similar skull shapes at the back. Holes at the top for muscles, holes at in the dinosaurs, holes at the side as well. And so we can map out where we think some of those muscles actually attached. Now we can do that on paper, we can make drawings, but actually what we really want to know if we want to understand this mechanism of levers and pulleys and muscles and tendons better is to get a 3D picture of what these look like. And this is where the X-rays come in. So, brief word about X-rays. We're all familiar with X-rays. I would imagine pretty much everyone, if not all of us in this room, have had an X-ray at some point in our life. So X-rays are high energy electromagnetic radiation, and so they pass through an object, and when they pass through an object, they attenuate, depending on the density of the material. And then the X-rays then strike a detector behind the material, which forms an image of the object that they've passed through. So this is a picture here of Wilhelm Röntgen, who was the person who discovered the potential of X-rays, and a picture here of, I don't know whether it's his hand or his wife's hand. I've read different things as to whose hand it actually is. But doing that test to see how the X-rays attenuated when moving through the hand. So yeah, we're all familiar with X-rays. So you may have had a dental X-ray where you've basically got a fixed tube here, emitting the X-ray source here, and generating these images here. The X-rays are attenuating more on the dense material of the teeth, so the enamel cap and then the dentine structure that's inside. Or in the case of a CAT scan or a CT scan, so computer-aided tomography or computed tomography, that's what that stands for, where you've got the X-ray source and the detector which move around the individual who's on this flatbed. We sometimes call these flatbed scanners for obvious reasons. Now these will give you these kind of individual X-ray pictures, and for the case of dentistry and some many medical kind of uses, that's enough to be able to see what you want to see inside the body, inside the structure. But you can, if you create a series of X-rays through the structure, stitch them together in a process called reconstruction and make these 3D images of objects. So like the whole body here, or the internal organs, bits of the pelvis, and so on. So we can do this in humans, and we can also do this with animal material, and we can also do it with our fossils as well too. Now the thing with the fossils is that invariably they're mineralized, so they're much more dense than the human body, even more dense than your teeth usually. And so we need a much higher X-ray source, a much higher energy X-ray source to actually get those X-rays through the material. So we have an X-ray machine at the University of Bristol, like a number of many university departments and hospital departments have. And this is an individual standalone machine, and you can get a sense of how big it is. This is Liz, our X-ray technician. She doesn't normally sit inside the X-ray machine. Obviously, the X-rays are off at this point. And Liz herself is actually quite short compared to me, but you can get a sense of the size of the chamber there. And so here we've got the X-ray source, and behind Liz here is a turntable where we would put our specimen, and behind the turntable is the detector plate. So when we switch the machine on, we close the door. This is a lead-lined box, and the turntable will spin round. X-rays will pass through it. We usually take around like 2,000 X-ray images as the specimen rotates on that turntable behind. And that allows us to create these X-ray snapshots, these reconstructions, which we can then isolate the regions of interest and create 3D models. So on the top row here, we've got some lorikeets, which were, was a colony of these at Bristol Zoo. One of these animals passed away, and we were able to take an X-ray of its head. And so here's the head of a lorikeet. It's upside down, if you can imagine. Now, one really important thing when we make these X-rays, if you've had an X-ray at the dentist or on a CAT scan, you're told to keep really still. And that's obviously because, like a photograph, you're going to blur the image. We have to make sure that these specimens stay really still as well, too. So we have all these kind of quirky ways of trying to stop them from moving. And one thing we've found is really helpful is actually, if any of you are flower arrangers, you know the florist's foam, the green stuff, is you can cut that and wedge it in, and it's very low density, so the X-rays pass through it, and it doesn't really affect your specimens, but it stops them from moving really, really well. And we have loads of other methods where we've tried cotton wool and sand and all this kind of stuff. So we get all these series of X-rays, about 2,000 of these as the specimen turns around on the turntable. We can then do the reconstruction algorithm, which enables us to create these series of slices, which you can basically move up and down through the animal's head. So here what we're seeing is the bones at the back of the skull. There's a big hole there where its brain is. And around here is all the soft tissues of the animal's skull. And there's its beak at the front there. So we're kind of going up through the skull. And in this case, the bones are really white. They've attenuated the X-rays the most. And we're able to use software that works on looking at the grayscales of all the pixels in that image and pick out the grayscales that we want. In this case, the ones that are more towards the white end of the spectrum, and then isolate these nice 3D images. So this usually works really well for living material, because there's a big difference between the density of the soft tissues and the bones themselves. But the problem arises when we're looking at fossils. And that's because the bones themselves have been mineralized, and they're in a mineralized matrix as well, too. And sometimes the composition of the bones and the rock that the fossil is in are really similar, and it's really difficult to discern what's fossil and what's rock. That's why we need the high energy. But this is a really nicely preserved skull of a precursor of mammals that's about 225 million years old. It's about that big. It's from fossil sediments, which are from Brazil, and it's part of a project that we're working on with colleagues in South America. It's a really beautiful skull that's really important in understanding the origin of mammals. Here it is upside down in the CT scanner, in the X-ray image, and here's our reconstruction. And so the bones themselves are these sort of dark grey areas, and the even darker grey area that's all speckled is this red sandstone matrix which the fossil is preserved in. But that's not bad for a fossil CT scan, and there's still enough contrast there to enable us to pick out the bone from the rock and create these lovely 3D reconstruction that a PhD student of mine, James, is working on, where we're able to isolate out the different bones of the jaw and teeth. So that's all good. So that gives us like the 3D hard tissue models, which we can then, if we want to, put our three-dimensional muscles on. So What will be really useful, though, will be to understand three-dimensional muscles in living animals and how they actually work out from CT scans. And one of the problems here is if you look at the lorikeet up here at the top, all the soft tissue just looks like a big grey sort of mass. There's no distinction there between the different parts of the tissue. So what we can do, though, is do some quite neat methods that allows us to increase the contrast of the soft tissues when we scan them. So this is the skull of a grey, short-tailed opossum. This was a specimen that comes from Edinburgh, from a repository where lots of animals that are in zoos, that pass away, go to the repository in Edinburgh, and then we can access that material and use it for scientific purposes. So what we can do with these tissues is basically dunk them in a special bath of solution that increases the radiodensity of their tissues. So this isn't our opossum. This, if you can just about make it out, is the head of a long-nosed gar, a fish. So there's its eye, there's its nose. Oh, it's the front of its snout there. And this has been dunked in a bath of potassium iodide. And that solution is gradually diffusing into the tissues. And what that's going to do is help us increase the contrast of our muscle tissues in the scan. So you can get an idea of this from our opossum. So this is a scan of the opossum's head without pre-bath. And here you can see that all the soft tissue just looks like a big grey blob. On the other side here, after that, the opossum's then been taken and it's been dunked in its bath of potassium iodide. In this case, we left it in there for a few weeks. And you can see that the solution is diffusing into the tissues. So you can see the fur is starting to come out really clearly. And we're also starting to get some distinction of the different muscle bodies, even some of the bundles of the muscle fibers as well too. So what we're able to do with this is then use the same 3D methods to actually create not only 3D digital models of the bones, but also 3D models of those muscles as well too, to see where they fit in the skull next to each other, their relative sizes, their actual sizes, and also get an idea of their orientations as well too, and therefore the leverage that those muscles would have. So all of this data is really useful because we actually know a few kind of general things about how muscles work that we can apply to fossils as well, because these are kind of generalities about muscles that we probably, if they're in living animals, it's likely from what we know that we find them in fossils as well too. So all muscles that we see in vertebrates and in tetrapods, land-dwelling animals with limbs and digits, are formed of these bundles of fibers. And these fibers might be what we call parallel muscles. So this is one of the hamstring muscles. And this is just, it's parallel because it's a series of muscles that just run in single fibers. On the right here, though, is what we call a pennate, sometimes called a pinnate muscle, where you've got a tendon that runs through, in this case, the middle of the muscle, where the fibers are branching off to the side. And this represents one of the quad muscles, the rectus femoris of humans. And the difference here is that there are different amounts and different lengths of muscle fibers packed into these two different types of muscles. And it's the actual, with muscles, it's not actually the volume of the muscle, it's actually the cross-sectional area of those fibers is what generates and what constitutes the amount of force that they can actually generate. So the more fibers you have packed in, the bigger the cross-sectional area of the fibers, the more force that muscle can generate. And so don't worry about this. It's just to basically illustrate to you that if we know a few things about muscles, like, for example, how heavy they are, their weight, and their density, which seems to be pretty much the same for most muscles, and the length of their fibers, we can calculate something called their physiological cross-sectional area. And we know that there's a relationship between this kind of cross-sectional area of muscles, and if you multiply that by the amount of stress, the force per unit area that we know muscles can generate, we can work out how much force a muscle of that size can produce. Now, this value of muscle stress as well, we can test this in living animals. And we know the range pretty much of what living animal muscles can generate in terms of their force per unit area. So this is all right if you've dissected out a muscle, put it on the scales, you can weigh it, you get its mass. You put it in a little solution of mild nitric acid, and it breaks down the connective tissues. You can measure the length of those fibers. If we don't know that, or if we've got one of these digital models, we can actually instead measure the volume of those digital muscles, and we can make an inference on the length of the fibers as well, too. We have to do, with the pennate muscles, we do a small correction that accounts for the offset of those muscle fibers from the tendon. But the key thing is that using these methods, we can get a value of how much force the muscle can generate. So I want to give you a few quick examples of how we've applied that to different fossil animals, and then we'll come back to that question I posed at the start with me on the TV saying about how much force the giant Pliosaur could generate. So one of the studies we did a number of years ago was on this animal here, Erlikosaurus. This belongs to a group of theropod dinosaurs, again, these traditionally known as meat eaters, like T-Rex. This group, Erlikosaurus, is what's called a therizinosaur dinosaur. They're characterized by having kind of long necks, short heads, these ridiculous long arms and huge claws. They were famously kind of featured in one of the more recent Jurassic World movies. And they have these big bellies as well, but the shape of their teeth is interesting because they have these leaf-shaped teeth, which we know are characteristic of plant eaters, and so these things were probably eating plants. So Erlikosaurus itself, you can see the size of its skull here. It's only about 25 centimeters long. The whole animal was probably about 2 to 3 kind of meters, but some of the therizinosaurs might have got up to 9 or 10 meters in size. Some of them were vast. But this was quite an early study that we did with this animal. It's fossils from Mongolia. We were really fortunate to be able to scan it when it was in the UK. And from that, we were able to kind of create one of our 3D digital models. So my then PhD student, Stefan Lautenschlager, Stefan's now on staff in Birmingham, but for his PhD, he studied this animal. And so we took the specimen, created these digital models from CT scans, and then using evidence from other therizinosaurs, Stefan was able to create this kind of reconstruction of what he thought the anatomy looked like. And then we went through this process of identifying where we thought muscles might have attached in the skull and on the jaw, like I mentioned before with the crocs and with the other dinosaurs. And then using that, Stefan was able to take this and digitally flesh out some of these muscles and get an estimate of where they sit in the skull relative to each other and how big those different muscle bodies actually were. And these are some of his illustrations and 3D reconstructions here. Now, this is still a hypothesis, yeah? We're not saying for 1 second that this was exactly what this animal looked like, but what we've been able to do is kind of parameterize it a bit more with our understanding of living animals, and then understanding what living animal muscle attachments look like, and then try and put this on the fossils. So, all jaws in invertebrates work as levers. And that's because you basically have a hinge, a fulcrum, which is the jaw joint, and you have an in-force, which is the muscle, the jaw closing muscle force, which is in red here. And then you have an out-force, which is kind of the reaction force where the animal's biting, the bite force. Jaws are third-order levers because the in-force is closer to the fulcrum than the out-force is. And actually, there's quite an inefficient lever system, because every time you put force in, you get less force out. But that's just how jaws work. If you think about it and try and imagine the levers in your own jaws working in a different way, it kind of can't place the muscles and the teeth in the right place, if you know what I mean. So for context then, we use this method then to, once we've estimated our muscle forces, to estimate how much bite force was generated by a contraction of these muscles at different points along the tooth row. And because this was working as a lever, the closer you are to the in-force, the higher the out-force gets. So if you bite at the back of the jaws, you generate more force than you do if you bite at the front of the jaws, basically. So it's really hard to measure how hard humans and animals actually bite. Because you can imagine, it basically depends, you can put a sensor in someone's mouth or in an animal's mouth and then encourage them to bite. But it really just depends on how bothered they can be to actually bite and how angry they are, how much you've agitated somebody to make them bite really hard on that sensor. And so the measurements for adult human bite forces vary quite widely. So I've taken a value here which is somewhere in the kind of middle to upper range, which is 680 newtons. So if you imagine that's someone biting really quite hard at the back of their mouth. So what we found for Elychosaurus in Stefan's work was that at the front of the jaws here, it could bite at around 50 newtons. So this is a picture of my older son here, and this is to represent that 50 newtons is about the same force that a teenager can bite at the tips of their jaws. So remember, the force is a lot less at the front than it is towards the back. So here's the midpoint of the back. This is my younger son here, who is now, would want me to tell you that he's now a teenager, but when he took this picture, he was a preteen, which is 75 newtons is about how much a preteen can bite at the middle of their tooth row. And then at the back of the jaws here, Elychosaurus is biting about 100 newtons, which is about the same bite force that a kind of 6 to 11-year-old child will be able to bite. That's the hardest bite that's been recorded for those. And I imagine if some of you have been bitten by a 6 to 11-year-old child, it's quite a hard bite. It hurts, basically. So that gives you a kind of sense of what this animal was actually doing. It's got a 25 centimeter long skull. This is how hard it's biting in kind of a human context. So it's going to be enough to be able to process, in some way, chop through some kind of plant matter, but it's definitely not going to be chomping through big bones. Okay, so the second example I'm going to give you before we come back to our Pliosaur is from a bit closer to home than Mongolia, which is where Elychosaurus is from, is the early Jurassic of South Wales. So here's a reconstruction of the early Jurassic of South Wales, but I want to point out these, you can see a little animal here which would have been about the size of a shrew. Now Kelly knows these animals very well because she studied them when she was in Bristol. So these are what we call mammaliaforms. So they're kind of right at the origin of mammals. So they have a lot of characteristics that make them look like a mammal, but they're kind of not what we would call in the crown, which is all living mammals and their closest relatives. And the early Jurassic of South Wales preserves fossils of these animals. And this one's called Morganucodon. There's another small related mammalia form called Cunitherium as well. It preserves these really well in a series of fissures. So we have some Carboniferous limestone platforms, and in those platforms, which represent kind of high grounds like the Quantocks and the Mendips and parts of south of Bristol, and then also parts of South Wales as well too. So if you can imagine the early Jurassic of this part of the UK, there's been a marine transgression. So sea level has risen, and it's isolated these kind of Carboniferous limestones as high grounds. And in those Carboniferous limestones are joints and fissures into which has fallen, been washed, the remains of animals and plants that were around in that environment at the time. So what's interesting now is that these areas have been, some are still mined for the limestone. And during the mining process, the quarry company find these kind of solid fissures full of fossil material within the Carboniferous limestones. And they look like these big, this is 1 centimeter, but some of these blocks are at the scale, some of these blocks are much bigger. And so you get this jumbled up mass of bits of bones, bits of plants, bits of insects that kind of look like this. But if you're able to break this down using some kind of, actually, because it's like a limey matrix, you can use like hot water and things, and it finally will sort of break down. Preserved in there are actually some really nicely preserved fragments of bits of these mammals and many other animals as well. But they're tiny. They're like less than 1 centimeter or so in size. So for this, we needed really high energy X-rays to be able to see something really tiny and to get really high resolution. So we use synchrotron radiation X-ray tomography. So this is electromagnetic radiation. that's emitted when charged particles travel in curved paths around these kind of what we call the donut here, so accelerated by magnets within the system. So you've heard of the CERN and the Large Hadron Collider. So this is a picture of a synchrotron which is near Zurich, called the Swiss Light Source. We've been there. We've also been to the European Synchrotron Radiation Facility, which is outside Grenoble in France. So nice locations to go and visit and scan some fossils, basically. So these produce high energy X-rays, which are able to reconstruct specimens at really high resolution. So you're in this nice environment. You kind of go in where we've got the Alps in the background, and you go into this kind of little hutch, and you don't see daylight for hours on end because you're basically putting these tiny little fossils into the X-ray beam, scanning them, putting the next one in, scanning them, and so forth. But what it does enable us to do is to take these little fragments of bones, and you can see how small they are here from some of these kind of mammalian precursors, scan them, get 3D models, and put them together in these 3D reconstructions to give us a picture of what some of these jaws might look like. So this is really interesting in itself to get an idea of the whole structure of the jaw. And we can look at similarities and differences, and if you're used to looking at these animals, you can spot things like the back of the jaw is much higher in Morganucodon than it is in Cuneiform, and this is the area where lots of jaw closing muscles attach. So you might predict that this has got more room for jaw closing muscles and things. In this reconstruction, we've chopped the teeth off for a reason that we did later on, but also we can see the teeth are actually different shapes as well too in the things. So we're able to take lots of material, scan it, and stitch together these 3D reconstructions. But then we're able to take this further and actually think about how these jaws might have worked. So in this case, we again looked at where we thought the jaw closing muscles might have attached, and instead of working out the force of these muscles, what we did was looked at the leverage that the muscles had in one jaw versus the other. And what we found was that Morganucodon's much more efficient in transferring that muscle force to bite force because it's got more efficient leverage in the jaws, but Cuneiform's got faster moving jaws. So there's a couple of differences there in their function. Another thing we do a lot of in the lab that I haven't really mentioned today is take these digital models and use engineering analysis to look at how they respond to loading and how they stress and strain and deform. And we use a method that's common in engineering and orthopedic analysis called finite element analysis. This method enables you to create 3D models of structures, and you can give them material properties like bone stiffness, and then run simulations to see how those structures respond to loading. And we were able to do this for our fossil jaws. So we simulated the muscle forces generating a bite of 2 newtons. So remember, a Lycosaurus was biting at 100 at the back of its jaw, but these things are like that big. They're like kind of large shrews. Of 2 newtons at the back here. So, but 2 newtons is what we know it takes to pierce through a beetle cuticle. And you can see that the jaws of Cuneiform experience a lot more stress than the jaws of Morganucodon when they're generating the same amount of biting force. So again, another sort of functional difference that we're able to pick out. And finally, what we did with these teeth as well too was we were also looking at clues about fossil diets from the teeth and the tooth roughness. So all of your enamel's pretty stiff, but it does scratch if you eat quite crunchy, food with kind of, if you imagine you're eating something that's got kind of little particles or stuff, it will leave kind of tiny microscopic scratches and pits on your teeth. And we're working with some colleagues in Leicester and looking at these kind of surface maps of the tooth. So this is the really high resolution map of the enamel, and the red areas represent peaks, and the blue areas represent troughs in the tooth structure. And in our 2 fossil mammals from Wales, there's some differences there. You can see Cuneiform's got a lot smoother teeth. And we're like, well, that kind of ties in with the fact that it's got faster moving jaws, its jaws aren't as strong, and it's not eating stuff that's as crunchy. And then we went a bit further than this and looked at these tooth textures and compared them to tooth textures that we find in modern bats, where we know what these animals are eating. We have colleagues in Bristol who study bat ecology, and they'd got bat specimens, they'd done stomach content analysis, and they knew what was in the stomachs of these animals. And we found some similarities in surface texture, particularly with one of the mammal organeucodon looked quite like these great horseshoe bats, which eat lots of kind of beetles and crunchy food in their diet. And Cuneiform, with that softer tooth texture, was more like what we see in brown long-eared bats, which eat more sort of softer foods like moths and things. And so we were able to do this kind of quantitative analysis of looking at tooth textures and looking at the variation in tooth textures. So the different coloured regions here represent the kind of a representation of the variation in tooth texture for the different living UK bats. And then on top of that, we have our fossils here, Organeucodon in the triangle and Cuneiform in the two types of crosses. And Organeucodon, these triangles plot in this region here where we find the great horseshoe bats that are eating crunchy stuff. And Cuneiform plots in the kind of opposite end of the plot here, next to things like the long-eared bats, which are eating much softer things. And in the fissures, we find little beetle carapaces. We find not moths, but scorpion flies, which are a bit softer as well. So, there's foodstuffs there that these animals might be eating. So you can see how doing this analysis kind of takes us through from, the bones and teeth look different. The muscles are working in a different way. The jaws are stronger or weaker than each other. The teeth are suggesting that they ate this thing rather than this thing. So it's a kind of, lots of different ways in which we can get at what these fossils were doing. So finally, let's return to our Pliosaur. Here it is in all its majesty. And what we're able to do with this specimen is too big to fit in any of the CT scanners. For a previous TV show, we tried to scan an ichthyosaur, another marine reptile skull, a scanner, and it's simply the X-rays just could not get anywhere near the specimen. So instead, what we use here is a surface scanner, which uses white light to capture the outer 3D shape of the fossil itself. So we can get a 3D model. We don't know what's inside, but we know the shape of it on the outside. And so this is my colleague, Andre, in Bristol. He was in the video at the start saying about Pliosaur being T-Rex of the seas. And for his PhD, he spent a long time scanning dinosaur skulls, basically, using this method. So he's very proficient in this method. What he was able to do then is you get these kind of images of the surface. The software we use stitches this all together to create this 3D model. From the 3D model, then, we can actually take some measurements of the cavities at the back of the skull where we know the jaw-closing muscles will have sat. And then using those methods that I talked about, we can make an estimate of how big those jaw-closing muscles might be. Now, pliosaurs are interesting because we don't have a great understanding of what they're really closely related to, and so we don't really know what their muscle structure actually looks like. But in this case, we took the fact that this back of the skull was just full of muscles. We took an estimate of the surface area of these muscles, and then we did a series of calculations to work out how much force those muscles could generate. So don't worry, I'm obviously not expecting you to read all of this, but what this represents is a series of different permutations where we looked at different possibilities for how these muscles might work. So, for example, here we've got our estimates of muscle force here. This is the magic number here. And this is if we take our surface areas as is, and then we look and see maybe the fibers were a bit shorter, and that means that they can generate more force. Here we look and say, well, maybe the fibers were a bit shorter, and perhaps the muscle stress was also different as well, then they can generate more force. We know that using this method where we just estimate the muscles from the size of the skull actually underestimates real muscle forces. People have done work in living animals. So what happens if we correct for this, and then we're getting a bigger value here? What happens if we know that we've probably underestimated again these muscles, and we kind of correct for like 10% underestimation? And then what happens again if we do all of these things, increase or decrease the length of the fibers, increase how much force those muscles can generate, correct for the fact we're just measuring the back of the skull, and introduce a bit of error as an underestimation. Then we get to this value here. So at the back of the skull of around 32,500 newtons. So that's a lot bigger, obviously, than our Erlikosaurus, than our human or whatever. But we're looking at a skull that's over 2 meters in length, and muscles which are probably this big, basically. But you can see how we've gone through a series of inferences there, where the real value will sit somewhere in this range here. And even the lower estimates here are kind of 10,000 newtons, which is still a pretty high amount. That's simply the volume of the muscle that's there. So if we compare that to what we know from living animals, that value in our Pliosaur is about 64 times the amount of force that a human can generate. And it is about twice as much as a great white shark can generate. We haven't actually measured a great white shark. People have actually, no one's got that brave yet, but people have actually looked at the muscle structure, the leverage of the muscles, all those things I've talked to you about to make an inference on how much force. And that number kind of seems about right. But here's T-Rex. Estimates have shown that T-Rex can generate a bite force, again, by working out how much the volume of muscles in the back of its skull, about 80 times that of a human. And if we look at some more familiar terrestrial animals, largest saltwater crocs and American alligators, about 26 times that of a human, so less than our Pliosaur here. This is a colleague of mine who works in Florida. He spends his time getting close to really big crocodiles and trying to get them to bite on this kind of force transducer here, which then sends a signal which picks up the amount of pressure that's been applied at the end there, so you can get an idea of its bite force. Hyenas, about 9 times that of a human, and lions, about 8 times that of a human. So that gives you then hopefully a kind of picture of, when I said on that TV show the amount of bite force the animal could generate, how we've kind of gone through that chain of kind of inference and hypothesized how much bite force that animal can generate within those parameters that I've just talked to you about. So I'll just finish there and kind of say, if I can, there we go. Hopefully you've seen how we can use X-rays and engineering methods and with dissection and microscopy to understand a lot more about living animals as well as fossil animals as well. Those two things naturally go hand in hand, because without the information from living animals, we're much more into the realm of guesswork with our fossil animals. But with those, we can kind of narrow down a little bit and make some constrained estimates on what fossil mammals, what other fossil animals we're eating, and how they're actually going about doing that. So thank you very much for your attention.